It's a pleasure to be here, as usual, and among friends, I see. So Dad already said my title. And really, this topic I want to discuss today breaks down administratively into the work of two excellent PhD students in computational biology, Mike Kramer, who's also getting an MD. So he's about to graduate, but then he has at least 2 more years and probably many more on the MD side. Why you'd want to do that, I don't know, but he'll be very powerful, I'm sure. And then Mike Yu for the second part of my talk, who's somewhere in the middle of his PhD. But Mike Kramer really talked about the descriptive, or did work furthering the descriptive side of this challenge, which is how do we factor the cell efficiently into a hierarchy of parts? And how do we do that from data as opposed to directly or indirectly from literature? And then Mike Yu gets into how do you functionalize this structure, and that'll be the second half. Another way of viewing this, though, is really 2 faces of the same challenge in computational biology and genomics. So the genomics view is that we now have gobs and gobs of genome-scale data. As you know, you can represent those usually as either a profile or a matrix or a graphical model or a network. And we have lots of ways of analyzing these data, usually related to clustering and classification. But these genome-scale data, we suspect, are not only modular, that is to say a list of clusters, but they are also hierarchical and multi-scale, and may contain far more information about cell structure and function than our current, say, round of clustering algorithms. That's challenge 1. The flip side of this is with respect to whatever one might elusively call a whole cell model, sort of this desire to really understand the complete structure and function of the cell, where I think we'd all agree we still need a set of approaches to do that, namely to capture structure and function of a hierarchy of cell systems. In my talk, with regard to that hierarchy, probably the most complete descriptive hierarchy we currently have in the public domain, and that's widely used by everyone, is, of course, the gene ontology. But as we know, this is not directly tied to data. It's indirectly tied to data through manual curation, and it's manual. It's not automated, by and large. And of course, being descriptive, it does not address quantitative or predictive aspects. So let me talk first about the descriptive aspect, and then I'll get later to the predictive aspect. So descriptively, it's long bothered some of us that networks are only really an abstract model of the structure of a cell at best. They could, of course, be very useful, but it's abstract. And by way of meaning, what I mean is if you open up the hood of a cell, it's not a wiring diagram like that. Very different, for instance, than if you open up a Pentium chip, there's an actual wiring diagram in there. And many other systems that are modeled by networks, there is an actual wiring diagram in there. In the cell, there is, of course, not an actual wiring diagram in there. If we look at what the actual structure is that this network represents, I've canned this to correspond to all the subunits of the proteasome, which, as you may know, is sort of the vacuum cleaner for proteins in the cell. It sucks up proteins and degrades them. Here's the actual structure is determined by millions of X-ray crystallographic images and computational methods that have been worked out over the past 50 years to integrate those. And you're looking here at sort of the side of this vacuum cleaner tube. So it's sort of a side view. You can see there's multiple folds of symmetry, but there's also, I want to point out, rich hierarchy. So here in red and orange, we have the so-called core and how it factors into subunits, at least structurally. On the outer edges of this thing, or ends of the tube, we have the so-called regulatory particle or aspects of this in blue and purple. The color coding I've made correspond over here. So you can see if you were to apply a clustering algorithm to the network or the corresponding profile that underlies these data, you might pull apart into 2 clusters, the red and orange, apart from the blue and purple. And maybe there's structure beyond that, but at least by eye, it's very hard to pull it out. So one question is, can we push the analysis further from clustering to, if not get at the structure, maybe that's eventually possible through some type of data integration scheme. But at least for today's talk, can we at least push it to see how much we can reveal about this hierarchy of subsystems and subunits. And already for just the proteasome, we have at least 3 layers of hierarchy here. Now, as I mentioned, of course, the current state of the art in hierarchies of the cell is the Go. We all use it. Most of us like it to a certain degree, although those of us who know something about a particular biological process often criticize it because we say, oh, they got it all wrong here, or they got it all wrong here, or it's incomplete here, or what have you. But nonetheless, it's I think all of us would agree been incredibly central to the discipline of genomics and bioinformatics. And so what I think they got right is this notion that we should factor the cell in a systematic sort of way into its component parts. And so here's sort of, if you started at the proteasome core complex, there actually are a couple of layers of hierarchy below here, but if I simply click on this at the EBI and get what's above this in the ontology, of course you get this rich hierarchical structure. So a couple of years ago, I guess 3 or 4 years ago at this point, Mike Kramer and Janusz Dukowski in my lab began to look at whether or not there was first of all, how close is the correspondence between the literature-curated hierarchy and hierarchies one can obtain directly from omics data. And then further, having established this kind of correspondence, went on to show that you can recapitulate much of the known Go hierarchy directly from data. Through essentially a glorified clustering procedure that I'll get into in just a second, but the idea is if you agglomeratively put dense interaction networks together, you can begin to define a hierarchy of clusters, and the question, of course, is whether those correspond to Go. An early indication that you would have this correspondence was this figure right here, where we simply took in yeast now, and I should say, or I guess apologize, that everything in my talk today will all be Saccharomyces cerevisiae, simply because if you want to study this problem fundamentally, there's far more data, both physically and functionally, for yeast than for any other species, without question. So here we're looking at physical or biochemical protein interaction networks. In purple, genetic or synthetic lethal interaction networks. In green, simply wiring together pairs of proteins whose RNA levels are co-expressed over many conditions, and all that in comparison to randomized networks. And we're simply taking the Go structure for yeast, the so-called Go slim for yeast, if you guys know about that sort of thing, and ordering the terms in it by size, which is roughly like a layering of the Go, if you think about it. This is sort of the lowest levels of the Go in terms of a hierarchical point of view, and this is near the top of the Go. There's about 6,000 genes beneath the root of this Go, so that would be the upper limit of the Go. And what you can see is that for very small terms, you have actually quite striking enrichment for physical interactions. This is a log scale here, so that's at least 50% of gene pairs assigned to terms of size less than 16 have already been documented to physically interact using systematic interaction mapping projects. So that's to me, that was a large number. And the second point here is that this enrichment persists not just for lower level Go terms or small Go terms, like small protein complexes, for instance, but medium size or medium layer Go terms, like the ribosome, which might have 50 or 100 subunits. And even, as you'll see compared to random, the largest Go terms, like organelles, mitochondrion, for instance, might be a Go term represented as size of 1,024. So this was encouraging because it told us that maybe there wasn't just the correspondence, but you could actually put Go aside for a moment, reconstruct the hierarchy, and then get most of what's already in Go and has been, in comparison, painstakingly curated over 10, 15 years. So I already mentioned to do this, we use a glorified form of clustering. As you know, there's lots of hierarchical or agglomerative clustering algorithms, but by and large, nearly all of them form binary trees as a result of their operation. And so the idea is you greedily pick the most two similar objects. In this case, we're again in the space of protein interactions, genetic interactions, co-expression, and other kinds of systematic data one can gather to create essentially gene-gene similarities among all yeast genes. And you can greedily cluster them to form a binary tree. But if one looks at Go, Go is not a binary tree, it's a DAG. That is to say, there are terms in Go, and you can see here implicitly I'm likening a join in this binary tree to maybe a term in Go, with the difference here that in the DAG, you can have terms that have more than 2 children and more than 1 parent. So, yes, Mike?
Embarrassing, I says, why is the random, in the previous slide, why is the random flat? Why does that go?
I think it just has. I think it really just comes down to if I randomly choose 2 protein pairs. Now, this has been about 4 years since I published this, but if you look, it's basically if I randomly choose a pair of genes, then that's the background chance that they would interact. Essentially. Okay, so the glorified clustering algorithm that we had to develop does really 2 things. It allows for multi-way joins, and it allows for multiple parents. And I think that there may be a class of algorithms out there that have not yet been described that could do this better. But that's the sort of 2 features that I think one needs for this class of clustering algorithms in order to get structures that aren't merely computationally convenient. The binary tree is merely a computational requirement of the algorithm or byproduct, but are actually structures reflecting the structure of a cell. That's the real goal here. Now, having done that, now we have a data constructed hierarchy versus the literature curated gene ontology. And note that Go has not been used to construct this hierarchy at all. However, having constructed the hierarchy, one can now use it as a reference against which to align the data-driven object. Much like when you first sequence a new genome and you run your gene finding algorithms on that genome, you at first have a bunch of unknown ORFs. You label them ORF1, ORF2, ORF3, and so on. By analogy, we do the same here when we first construct a new data-driven ontology of a particular set of data, here the yeast cell. We at first just name these term 1, term 2, term 3. They're orphan terms, if you like. But now through alignment against a reference standard, just like you would align your genome and your ORFs against a reference, which would be GenBank or something, and every time you get a good sequence alignment and the aligned sequence in the database is named, I just found actin, I just found what have you, my favorite gene, based on the sequence similarity, you can now transfer all English language knowledge over to that formerly orphan gene, now no longer orphan gene, and name it and so on. So we can do the same with the gene ontology alignment. And here we didn't invent any new algorithms, but there's already, it turns out, a collection of ontology alignment algorithms, not in bioinformatics, mind you, but in other disciplines of computer science that we could avail ourselves of. And just briefly, the way they work is they compare labels on the terms. Here the label is just the list of genes underneath each term. So we compare how close the labels are and have a score that incorporates that. But also in sort of an EM-like fashion, you look at how good those alignments were for your children and your parents and simultaneously come up with a one-to-one mapping, or at least that's the idea, is to come up with a one-to-one mapping or a 1-to-0 mapping. You can simply fail to align a term, but you don't allow 2 terms to align to a term, for instance, in the other ontology. So having done that, we can now begin to name some terms and recognize them in our data-driven version, as well as recognize which terms are novel and which terms in Go we missed. But first, before I get to that, just to give some intuition about how this thing works. Here's some unpublished data from Mike Kramer's latest project. He's trying to publish it right now, where he's taken a case study of one particular process that's in all eukaryotic cells, and certainly in yeast. It's called autophagy. It's the process by which cells digest other components or components within the cell, including proteins. It's been linked to stress, aging, cancer, and lots of other diseases, Alzheimer's, for instance. Here are some of the core autophagy genes in the middle, ATG 27 through 24, as the name would suggest. That's autophagy. And then as outgroups to show you what dissimilar genes look like, we've put ARF1 here at one at the top and BCK1 at the bottom. And what you're looking here in terms of the heat map is an integrated gene-gene similarity matrix from those protein-protein data, the genetic interaction data, and so on. So don't worry so much about how we got that integrated gene-gene similarity matrix. It's a straightforward thing that lots of people have described how to do to integrate multiple data sets. But you see in the gene-gene similarity space, if you went to cluster this into a discrete flat set of clusters, you'd run into problems and have to make some arbitrary decisions. So for instance, you would say, well, ATG 3 and 7, that forms a tight cluster. Okay. But 7, 12, and 10 also form a tight cluster, and 10 and 27 also form a tight cluster. So clearly we need to assign genes and also terms to multiple parents in this hierarchy. And then we really want to preserve the whole hierarchy itself and not have to cut this thing anywhere, because you can see a larger but weaker cluster here all the way from ATG 10 down to ATG 8. So if you see in the hierarchy, and having now built it and just aligned it directly against Go, we can now begin to find these structures and begin to name them, again, from the Go alignment. So here for ATG 7, you can see here it gets assigned to 2 parent quote-unquote terms, 1 together with 3, and 1 together with 12 and 10, and so on and so forth. And then this entire block gets called core machinery of autophagy. It actually ends up aligning against a Go term called core machinery of autophagy. Now, I mentioned there's orphan terms, that is to say terms that aren't in Go. Here's one of them. We had to just leave it labeled term 183. It's potential new biology. Maybe it's a new protein complex. We'll have to find out. And then the red bordered nodes here are terms that we have now named but weren't originally in Go. They now are being put in Go. I believe they might already be in Go. We have to look by our request. And the reason Go put them right in is because we already found several papers supporting the creation of each of those terms. So here's one where we believe from data there's a term, there's no paper supporting it, so Go won't put it in. Here's a case in these 3 cases here where there were ample literature already out there, and so we simply sent the papers to the Go editors, and they viewed them and put these terms in. So hopefully that gives you a feel for how you begin to build these hierarchies and annotate them from data or from the Go. And if you zoom out and look at the whole hierarchy you build for a yeast cell, this is one rendition of it. The root of this hierarchy is up here. You might observe, I promised you a DAG. This is actually a tree. It's not a binary tree, but it is a tree. Simply to view this whole thing, we had to cut multiple parents. So we had to choose the most important parent, unfortunately, whenever there was more than one. But nonetheless, it lets you lay out the whole thing and talk about it. And so you can see here at the highest level what happens is you quickly, based on data now, trifurcate into a mitochondrion membrane set of components and the intracellular compartment. Those get labeled directly by alignment to Go. That reflects what's in Go. And I think it is remarkable that just based on sort of protein-protein and genetic interaction and co-expression similarity, one can pull apart and cluster out components at this high of a level. I think that is kind of interesting. And then notice that most of the other labels that get put on different parts of this structure, again by alignment to Go, come down here in the intracellular compartment, probably reflecting the simple fact that we've studied as a field much more closely what's inside this intracellular compartment than what's at the membrane or in the mitochondrion. Now, zooming further, if you recall, I showed you the structure of the proteasome. Let's look at what the proteasome looks like in now the ontology that's been constructed, and that's over here. Recall the data were of this form, if you visualize them as a network. But if you process that data as an ontology, here is that region of the ontology. That's up towards the root there. So notice, again, I only put labels on terms that have been recognized and aligned to Go. So in Go, we found the proteasome complex. We've factored the proteasome complex into that core and regulatory particle you saw in the structure. And in turn, what you couldn't even see so closely in the structure was the core itself. Well, I guess it was the orange and yellow or red and orange, I believe, subunits there. We have an alpha and beta subunit the core factors into, and similarly the regulatory particle factors into a base and a lid, and then maybe you have some other aspects of the proteasome here that aren't exactly what's in Go, and whether those are errors or novel discoveries is sort of now is the opportunity. The opportunity arises that you can look into that.
Trey,
Ron.
which of the three Go hierarchy are you aiming to?
Excellent question. So there's these three Go hierarchies: biological process, cell component, and molecular function. Actually, it's a good question because it's my next slide. So we aligned against all three, and this view is with respect to, so Nexo is what we call this data-driven ontology, network extracted ontology originally, although as you'll see, Mike Kramer has renamed it. So simply looking in terms of the 1,402 terms in Nexo, how many of those are corresponding to a Go biological process, cellular component, or molecular function term is shown there. But to me, more revealing is to look with respect to Go, what fraction of Go terms in each of these three ontologies we recover from data. And here it's very clear. So here it looks like maybe it's a biological process ontology, but the problem is there's far more terms in the biological process ontology. So in terms of coverage of Go, you're at 60% roughly coverage of the cell component side of Go. This is an unsupervised approach. In just a bit, I'll show you how Kramer has now tried to turn this into a supervised approach so you can target particular ontologies if you like. But here in this, all the vagaries of whatever decisions were made here, we got the cell component. Now, more recently, a few years ago now, I'm now marching forward a year or two in time, Mike, Vineet Bafna and I worked together to try to improve the algorithm, and this is more just an advertisement for this paper. I don't have time to go into the details, but just if I had to give you a high-level novel aspect to this paper versus the earlier work, it's that it's more error tolerant, so we can add noise both for false positives and false negatives, and the precision and the recall are much more tolerant to that error. And so now you'll see sometimes this word Clixo, which also reflects a slight change in the way the algorithm works. This is called clique extracted ontologies, and it's actually inspired from some of Rodet and Ron's work from way back, where they use sort of clique detection as a way of clustering, and this is sort of hierarchical clique detection, if you want to look at it that way. Okay, so we now are very excited to use this approach to begin to describe a variety of ontologies, and the question is, how does one begin to use this in your, say, experimental laboratory as a way of pushing forward the biology, sort of in an active learning sense, if you like. And so this slide partially summarizes what I've said. Maybe now is sort of a template for how one might apply such a pipeline, and also how one might do it in a systems biology cycle or in an active learning sense. So the idea is you choose your favorite process. In this case, for Mike Kramer, it's autophagy. You try to build a hierarchy of that process using public data, as much public data as you can get your hands on. You integrate those data to derive a gene-gene similarity network, and using this clique extracted algorithm I described, just alluded to, you get a hierarchy, or a DAG, which reflects the structure of that data set. Now, typically, by the way, when one begins to study a process in experimental biology, one doesn't incorporate prior knowledge, by the way. One just says, I'm going to go do a big genetic interaction screen for my favorite process. Why? Because I can. So we think one important change that needs to happen in the field moving forward, and I'm now sort of addressing experimentalists, right, is take advantage of all the data we've already generated as a field to inform your process as much as you can before you start doing experiments. That's point one. But now having built the best model you can, you've aligned it with Go to see how much of what you of the public data sets are already reflected in Go, and how much novelty there might already be. Maybe you're done. Maybe you already find a novel process or subcomponent here that you want to study. But if you want to study more, now the key would be, can you go back and look at these data sets across all the different omics types of data and do some kind of experimental guiding or selection as to which new space of experiments might be most fruitful for building this ontology. By the way, the entire ontology for autophagy wasn't just that core I showed you before. It looks like this. I'm not going to go into all the detail here. Here's the core componentry here. But just to point out, even for a modest process of, say, 100 to 200 genes, these things get quite complicated. Nonetheless, when you sit down with your cell biologist friends, and here we're working with the Subramani lab, an expert in autophagy, we find that they really find these diagrams intuitive, and I'll show you an example of some follow-ups they've done in a second. But first, let me address this experimental design component. So having built this ontology and aligned it against Go, one can now use that Go alignment as sort of a benchmark to talk about how performant a particular data-driven ontology is as you erode the different data sets underlying it. So when I say fraction of maximum performance, what I really mean here is one is essentially the accuracy with which I'm recovering the Go branch of autophagy with all the data. And you can look then at how you decrease that fraction as you pull out data. And we've looked at multiple ways of pulling out data. You can pull out interaction by interaction, and qualitatively the results that I'm going to show you don't change. But here we thought it was a little bit more intuitive to pull it out study by study. So if you look at all of the data that are in yeast that inform protein-protein interactions or genetic interactions or co-expression and what have you, and I should say we simply borrowed Ed Marcotte's approach for doing this. He has something called YeastNet, if you've heard of that. Essentially, our data integration scheme is just YeastNet. And we freshened YeastNet from whenever the paper was last published. We added a few more data sets, but the algorithm is the same. There's, I think, 80 to 100 different studies, quote unquote, that they summarize in that work. And so we can pull them out one by one according to their different types. So there's studies of genetic interactions, protein interactions, and so on. And so what you can see here is this thing is somewhat sensitive to pulling out either genetic interactions or protein-protein interactions. There were only 10 versus 12 of those in this integrated data set. Think about them as papers. But there's 50 co-expression papers. Interestingly, you can pull them all out, and it has no effect on your ability to reconstruct the ontology. Okay. So based on this, and there's maybe other ways we could have viewed. And the other point that can be made here is you can look at the absolute reduction of performance, but you can also look at the slope. And so based on the slope, even though pulling out all protein-protein interaction papers was slightly worse than pulling out all genetic interaction papers, the instantaneous value of a single new genetic interaction paper seems slightly more valuable. But again, you're really splitting hairs here. Both of these data sets appear quite valuable. And combine that with the fact that my lab has already the experimental pipeline to do genetic interactions and not physical interactions. It was an easy decision for us. We went in robotically and generated a bunch of new genetic interaction experimental data. Now, importantly, even though Charlie Boone and Brenda Andrews and others have covered much of the space, and I think this afternoon we're going to hear about how they've covered even more of the space of genetic interactions, at least in terms of what's available to me right now, it's only about 30%. So we sought to fill in the other 70% for autophagy. But the other key point here is we did it not just in untreated conditions, but because of the fact we're studying a process that has known stimulants, we could look at how those genetic interactions change across autophagy relevant conditions. And so that's what's done here. We have sort of the untreated, we have the untreated profile, which I'm guessing Anastasia is going to just show us later. That's probably just a duplicate of what she now has in the mother load, okay? But then we also have starvation, rapamycin, and the differences between all of those profiles. So in all, we considered this either 3 new experiments or 6 new data sets, however you, with some covariance between them, but however you want to view it. And so now we can look at how much those new data that have been targeted both in terms of the gene set and targeted because of the conditions being relevant to our process influence our ability to reconstruct autophagy Go. So just to summarize, here's what we had done with all prior data in the first iteration of the systems biology cycle. And this is just recapitulating something I showed you earlier. Again, looking at how well we recapitulate knowledge in Go. Here, by the way, you can this measure is reducing Go itself to a gene-gene interaction network using this ResNext semantic similarity measure some of you guys may know, and then just looking at how our edge scores correlate with theirs after, either before or after integrating with the new data. And you can see that what's interesting is just looking at the gene-gene similarities, just using our new genetic interaction profiles alone, do better than all prior data, a little bit. And so I think if there's an explanation, it's because we're targeting towards this process as opposed to all prior data, which largely does not target this process. But then, of course, when you integrate the new data with the old existing prior data, then in fact you can do even better. And so overall, as a result of going one time around the systems biology cycle, we now have a hierarchy that correlates with Go at the 50% level as opposed to the 30% level. So we think that's a substantial as well as significant change. Of course, if you shuffle our new data, then you get no correlation, just to perform that negative control. And I mentioned that our collaborators find this a particularly useful structure to sort of look around to get new hypotheses for experiments to do. So just to show you one of these, there's a number of things ongoing. In the prior version of the ontology constructed without the new data set, they have this favorite gene that they study, ATG26. It was assigned to essentially one of the highest level terms in the original version. Whereas after adding the new genetic interaction profiling data and building that new hierarchy, aligning against Go and so forth, ATG26 got placed much, much more specifically. I'm just clipping out a parent and a child term near the leaf of this ontology. So it got grouped specifically together with these three other autophagy genes, 19, 27, and 11, suggesting that maybe ATG26 functions in the term that those other genes were already annotated to, which was protein localization to the pre-autophagosome. To prove that that's probably the case, or at least to provide evidence that's probably the case, they tagged the known pre-autophagosomal gene ATG19 with GFP, looked at how it overlapped with the localization of ATG26, this new or a newly assigned protein with another fluorescent dye. And what they're trying to show you here is that these two components do not overlap in untreated conditions, which is probably why they've been missed in interaction screens so far. But when you add rapamycin, notice the cytoplasmic general location of ATG26 sucks up into a punctate-like structure, and now these two overlap. And so that combined with a number of other experiments they've performed now lead us to believe that ATG26 is indeed part of this process. So I'm a little more than halfway through the talk, but I'm through the first part, and let me summarize the descriptive part of this talk. So maybe a overriding theme here is that when we look at these hairballs of molecular interactions, we're too close to the data. Maybe it's really more akin to trying to learn something about a protein structure or the structure of a protein complex like the proteasome by looking at these X-ray diffraction images. Certainly people do this and used to more in the past, but nowadays we have a robust set of computational algorithms, largely known by the name of structural proteomics, that converts millions of these types of images into one of these structures that one finds in PDB. So I think the exciting idea here is that maybe we're in the same mode with networks. We're really looking too specifically at the data. This is somewhat sort of like the raw data, or close to the raw data. And what we need is a set of computational algorithms that can reliably process these data, if not directly into the structure, into data-driven ontologies and other types of structures that inform overall cell structure and function. Incidentally, though, I should say, in the proteomics field, certainly not in my work, but in the proteomics field, if you hear Rudy Aebersold talk or any of those guys, there's now a big push in what's called chemical cross-linking, which is a way of measuring interactions not just among whole proteins, but among peptides in those pairs of proteins that Rudy and others think will maybe get you somewhat closer, at least, to structure. So that's the summary. Yeah.
I have a question. Before you move to another subject,
please.
I have a question about noise. So when you're comparing now the structural proteomics with the, let's say, genetic interactions, protein interactions, and so on, there is noise all the time, and you're extracting a
yes.
large number. And the more data you have, the less noise you have when you are doing the structural proteomics. However, we know that both the genetic interaction network and the protein interaction network is very noisy. It has a lot of false positives and false negatives. And so at the higher hierarchy, it's very easy to eliminate the noise or to disregard it. But when you go down in the hierarchy, then the noise becomes really important. I mean, it's very hard to see whether you have a new genetic component.
No, it's absolutely.
A lot of noise.
Completely agree. So,
The
but
question is, how do you deal with that?
well, I think the analogy is actually quite good still to the structural proteomics view. So the more data you have, the less noise. And so the original idea of people like Ed Marcotte, Mark Erstein, later Olga Troyanskaya, and so on and so forth, who integrate many lines of evidence to form gene similarity networks, is that each line of evidence may have a certain noise characteristic, but that noise goes down as you integrate more and more lines of evidence over these different kinds of networks. So certainly that's a key part of this infrastructure here. And in this active learning sense, the idea is to target new data sets, in a sense, to where you can clean up and make more stable some of those terms. That's also why Mike Kramer published that paper with Vineet Bafna at ISMB 2 years ago, is really an exploration of how noise influences this whole process. So check out that paper too. Not claiming we've had the final word on noise and this topic, but we have sort of thought a bit about it, and I think it's quite analogous to structural proteomics or to any situation. The more averaging you do over many data sets, the better noise characteristic you're going to have. Okay, so let me now, in the last, say, 15 or so minutes, flip to this second challenge that I mentioned, and this is Mike Yu's projects, which is that, networks and ontologies are mostly descriptive maps. Certainly Go has been used in all sorts of predictions, but there's nothing inherent about Go or the Go structure that's predictive. That's the point. However, by the way, we happen to be reading this paper about how Siri is constructed a few years ago. This is one of the last papers that the authors of Siri published before they got bought by Apple and stopped publishing papers. I think these days probably interest in ontologies has given way to deep learning and that kinds of thing. But nonetheless, it remains that when one speaks a command to agents like Siri, other agents, ontologies have long, as many of you know, been sort of a cornerstone of how we process a query to give an answer. And so, for instance, this is a piece of one of the many ontologies that these guys, at least now 10 years ago, had put in Siri. Who knows what's there now, but at least in an early version, this was one of the ontologies that Siri has. As you speak to Siri, Siri is trying to take your voice commands and slot those words in as states into one of the many ontologies it has in its lexicon, and somehow the most probable fit is then the one that it uses. And so and these are not my words, not in the original paper. If one said, Siri, find me a good sushi restaurant tonight for 2 in my city, La Jolla, this might be the best fit to that ontology. And so you notice what's happening here is the state of city now becomes La Jolla, the style becomes sushi, party sites becomes 2, and so on and so forth. And based on that information, Siri can now backpropagate and fill in terms like state and zip code, and up the ontology can return answers to your query like a list of restaurants that meet these state criteria. So the idea was, why can't we do something similar with Go? With Go, it's simply descriptively used. Here we have both predictions and description that are really being facilitated by ontologies. So with Roded Sharon's group, Mike Yu embarked on the following project, which just, by the way, came out in Cell Systems about a month ago. So the idea here is now to go back to yeast and address queries of a very particular kind, but which capture the general genotype-phenotype problem in genetics. And as you know, lots of problems in genetics, one might even say to a first order, all problems in genetics can be mapped to genotype-phenotype prediction. So again, yeast is a great model to think about functionalizing the Go because of those genetic interaction experiments that Anastasia is going to be talking about later. We have, even as of this publication, about 5 million genotype-phenotype pairs of the form a pair of genes is deleted and they measure a growth rate. Right? A genetic interaction experiment is essentially a genotype-phenotype relation. That's the point. It's a particular kind that involves disruption of exactly 2 genes. But we have lots of these pairs. In fact, it's by far the largest genotype-phenotype database that I'm aware of. So here's how we try to predict growth given genotype using an ontology structure in something akin to what I showed you for Siri and adding state to terms. I've now turned the ontology upside down, so the genes at the leaves here are up and the root is down below. But now imagine the game here is to add states to terms, which reflect essentially some information about whether the genes underneath those terms or here above those terms have been disrupted. So what one really wants in a general sense is for a term to have an activity state, just like we can say a gene knockout disrupts and abrogates the function of a gene. What does that do to the function of a term? That's the sort of set of latent variables we have here. One can imagine lots of ways one might try to update the state of a term or determine whether a term's been disrupted, given states on its children. Here we used a couple of really simple policies. We tried a simple OR gate. So basically, a parent term gets disrupted if any of its children are disrupted, and so on. We also tried a simply summing the number of genes assigned to a term that have been disrupted. But again, one could imagine in the future lots of different schemes that one could use to propagate function from genotype to phenotype here. And while I'm on that topic, the ontology really does provide a nice bridge between scales. So if you think about this, what's the scale of genotype? It's changes to nucleic acids, which are sort of sub-nanometer or nanometer scale, if you like. And if you think about phenotype down here, for a yeast cell, it's sort of overall growth characteristic of the cell itself, which is maybe 10 microns. 100 microns for a human cell. So that's the scale. So we're going from nanometer scale to tens of microns. Ultimately, one can imagine similar schemes for predicting, say, human phenotypes, which would be a meter scale. So really what the ontology here is doing, they call it the gene ontology or its data-driven cousin, is it's propagating perturbations at the nanometer or to objects nanometers in size, to objects tens of nanometers in size, hundreds of nanometers in size, all the way up to your final phenotype scale. So it bridges these scales in quite a nice way. Now, so now that we have some simple policy by which to set the state of a term, we can now translate a genotype into what Mike Yu called an ontotype. And an ontotype is the complete set of states on all biological objects in a cell, while genotype is the complete set of states on all genes. So ontotype includes genes, it includes phenotype, and all objects in between. Is the idea. And so here you can see how ontotype begins to coalesce different genotypes into similar or same states. So here as a toy example, as a simple one, if you disrupt gene B and D, from the point of view of the ontotype, that is the same disruption as a disruption of A and D. Why? Because A and B are equivalent. They are both assigned to the same term. Here a -1 means 1 disruption, -2 means 2 disruptions underneath that term. And 0 means no disruptions. Given now this ontotype, we now abandon any notion of a whole cell model and simply use straightforward classification and regression. And so now Mike used random forest regression to regress these ontotypes in a completely black box way against the known phenotypes in a cross-validation framework. So at least we can push genotype using some biological knowledge to ontotype, at least at the present point in time in our hands. After ontotype, it's complete machine learning. Now, why should this work? Let me show you a very different view of the same phenomenon. If one takes these genetic interactions, that is to say pairs of yeast genes that when disrupted lead to a slow growth phenotype, one sees striking similarity or correspondence to the Go or data-driven ontologies. So just to give you an example of what we saw that sort of motivated this idea, if one looks at genetic interactions, there are lots of genetic interactions between the set of genes shown on the left and the set of genes shown on the right. That is to say, delete pairs of genes across those two sets, and the cell doesn't grow very well. Now, in terms of the ontology, it offers a ready explanation for that. All of these genes are encoding kinesin. All of these genes are encoding dynactin. And earlier, papers from my lab and others would have called this between-term enrichment. But now that between-term enrichment actually becomes a within-term enrichment at the next layer up in the ontology, because, as you may know, both kinesin and dynactin are part of the microtubule-associated complexes. So with regard to the learning, all of these data would allow the machine to learn that one disruption here combined with one disruption here, or alternatively, 2 disruptions here would lead to slow growth. And that's the kind of transfer function we're going to try to learn. So how well does it do? If we simply look at the growth or interaction score predicted by this ontotype procedure, and look at in cross-validation how well it recovers the measured interaction score from Costanzo et al., we see pretty good correlation. It's about 0.35, I think, 0.3. So there's that. If we substitute the literature-curated Go for yeast with our data-driven version I presented in the first half of my talk, recall that was called Nexo, it actually doesn't do quite as well, but still both of those do way better than randomizing the ontology. The corresponding versions of the ontology as a negative control. And as some of you know, there's been lots of efforts over the past 10 years to try to predict synthetic lethals using a variety of schemes. Here we've compared against all of the ones we sort of have find as representative. Some of these are just simply newer versions of older ones, and so we didn't look at the older ones. We looked at the newer ones. But all of these prior approaches are non-hierarchical. They use features, in many cases, many features, but not organized in this way. What's really cool, I think, about the current approach is how simple it is. There's only one kind of feature, a term state. In contrast to some of these other approaches that have sort of thrown everything in the kitchen sink to try to predict genetic interactions. I'd like to also point out that if we flatten our ontologies by reducing the ontology using semantic similarity to a network, and then apply sort of network schemes, it doesn't do as well. So we think, in fact, it really is something about the hierarchical aspect. But I do think more studies are needed. And then if you want to look at this in terms of if you prefer the correlation, something like a precision recall curve, of course, if you look at sensitivity, specificity, all these approaches look fantastic. I'm not showing you the ROC curve, not because it's bad. It's just too good. And the reason it's too good, by the way, if you care, is this is a very unbalanced learning problem. So there's far more. The vast majority of pairwise gene disruptions have no phenotypic effect. Everyone gets it. Mike's like, we get it. Okay, so look at precision recall, and it really keeps you honest about how all these things are doing. Okay, so now one can begin to look back though and try to interpret the ontotypes or the prediction process. So just before I scale up to a larger example, let me focus on again this one. And just to see the color coding I'm using here. So as we browse around this functionalized ontology, we're going to look at terms that enrich for lots of positive or negative interactions. And so this shows that this term enriches for lots of both. And then we're going to look at how those enriched interactions factor among the children and children's children of those processes. This is the subsystems underneath that term. And so here's another good example. It's quite intuitive that gets pulled out. You find that oxidative phosphorylation enriches for both positive and negative interactions. But the positive ones all go this way. So keep in mind, a positive interaction is two disruptions that actually produce better than expected growth. A negative interaction is two disruptions that produce worse than expected growth. So here you can hit electron-coupled transport or until the cows come home, essentially, at least twice, and the cell's fine. Okay? But if you hit one here and one here, the cell's dead. And at this layer, you wouldn't have seen that. You only see that explanation here. And so now you can begin to see how that's going to convert into ontotype logic in the machine learning. Here's a bigger example. DNA repair, a process more near and dear to my heart. So here, DNA repair is a term enriches for lots of interactions, both mostly negative, but some positive. But then if one looks at the subsystems involved in DNA repair, double-stranded break repair, recombinational repair, mismatch repair, nucleotide, and so on and so forth, one sees the blue, the positive, pushes all the way down essentially to synthesis-dependent strand annealing. So you can hit this process. Essentially what's happening is one hit to this process has the effect on phenotype. Multiple hits don't matter. Whereas there's lots of semi-redundancy or synergy across pairs of DNA repair pathways, much like we might expect. But again, you can now see how all that logic can then be converted into a prediction of growth. Yeah?
What if you have an actual synthetic rescue? What is the logic of that?
So synthetic, can you define for me what you mean by synthetic rescue?
Two things that signally create a bad growth phenotype, you put them together and there is no effect on phenotype at all.
I mean, the problem is for us at this layer, that just looks like a positive. That gets treated the same as any other positive epistatic interaction, and we're not going to be able to distinguish it in the current framework. Now, I think what you've just done is made a suggestion for future work for how, if depending on the scores one gives this thing for learning, one could maybe learn those effects to be separate from straight epistasis. But the last slide I want to show here is I think to remind us here that this is really not. I focused a lot on prediction of genetic interactions because that's the data I currently have in large number for genotype phenotype. But just want to remind people this system is not restricted to prediction of just pairwise gene disruption phenotypes. It converts any genotype, simple or complex, into an ontotype, which is then used for prediction. So there is one paper we could found from Nevin Krogan's group that looks at it's a large-ish screen for three-way interactions. And I won't go into the details of that study, but basically what's shown here, just like I was correlating our predictions versus the measurements before, these are pairwise and triple deletions. And you can see here that what happened in their hands is the double deletions involving the gene CAC1 and any subunit of the HER complex lead to slow growth. There's 4 subunits of HER. That's why there's 4 points. but that was rescued by addition of another deletion at ASF1. So those circles go up to the squares where there's really no phenotype effect on growth. And so we were pleased to find that our system, without any training, maybe we got lucky, we probably got a little lucky, but nonetheless, in the one example out there, or a study of three-way interactions, we can predict that effect. And what's basically happening is shown here, breaking down the structure of the Go and the function that's then ascribed to that Go, and you can see how the prediction is made. Essentially, all of these genes fall under chromatin organization. All three of them are ascribed to nucleosome organization, but only two of them to DNA replication independent nucleosome assembly. And so what basically happens is, if you hit DNA replication nucleosome assembly twice, then essentially that cripples the cell so bad you can't observe any more phenotypes, and so there's no interaction prediction. But if you sort of hit it once in one knee but not the other, now it's crippled but still functional enough that you can reveal phenotypes within nucleosome organization, in which case 2 hits but not 1 to nucleosome organization kill you. So is this complicated? Yes. But did the machine learn that complication? Yes. So to summarize this last part of the talk, and by the way, so I'm showing off, I'm sorry, but we're happy with this. The editor said this was the most psychedelic image she had ever seen, kind of thing. But the idea here is that perturbations to cells at the nanometer scale to genes can be converted to observations of perturbation at the micron or meter scale, the level of cells and maybe eventually people. And one convenient structure for connecting or bridging those scales is the gene ontology, because it really captures a small aggregations of gene products in small protein complexes, larger complexes, pathways, organelles, and finally cells, in a way that can propagate or aggregate mutations from small funnels to large funnels, and so on and so forth. So like the first part of the talk, I don't think that the algorithms we're using now are the algorithms that will be used next year even, but I do think there may be suggestive of work that needs to be done and might be quite exciting. And so with that, I've already, I think, thanked hopefully everyone involved, or the key players involved, as I went along. Certainly, I'm thankful for key collaborations with the Bhavna Sharan and Supramani labs, and I'll be happy to take any questions.
For one of your earlier slides, you compared the cellular compartment, molecular function, and I forget the third one.
Biological process. Yeah.
And the cellular function seems to do a lot better than
cell component in the unsupervised mode. And later in the autophagy piece, I slipped in a supervised approach without explaining it. But was that the question you had, or was there another question?
Yeah. So why is cellular compartment so much better than the other two?
So in that study, so I thought a bit about that. I think it's just that you saw that. That these ontology construction procedures appear to be driven by protein-protein interactions and genetic interactions, and less by gene expression, at least in one way of analyzing it. And if you look at what is in the cell component branch of Go, there's a lot of protein complexes. Biochemical interactions maybe best predict cell component. That's my guess.
And is that also most useful for the genetic interactions?
Yeah, and so if you recall, when we were predicting genetic interactions, that is to say genotype from phenotype or phenotype from genotype, we tried both the data-driven ontology and the Go, and they worked about the same. The Go worked a little bit better, but the data-driven ontology, again, is based on the same kind of information. I do think the way forward here, just like we always have structure-function relationships in biology, I think the way forward is to build the Go structure from structural data, so not genetics. Originally, we threw it all in. Now I think that might have been a mistake. I think maybe what we should do is keep the structural information separate, build the hierarchy, and then functionalize that hierarchy at a later step using pure genetics. That's not what is done here. Well, certainly in the last part, we had to show that we weren't predicting genetic interactions just because we built off of genetic. But having done that, we weren't quite as pure about it as I think people should be.
Yeah.
Oh, and then Teresa, yeah.
So to follow an earlier question, do you think it's feasible for you to take into account the level of noise when you put different information together?
Yeah, so there is. So in this Clixo paper, that was what that paper is about. There's one parameter called alpha that you can use to dial your error tolerance. I'm nervous about this parameter, by the way, but there is a parameter.
But for the ontology, do you think it would be good to put in that kind of indication of?
Yeah. So actually, hey, and I forgot one thing. So let me show you what we're building. I have to merge my displays here. Figure out how it works. So we're trying to come up with an online version of these ontologies that can be browsed. So here's the one that Michael Kramer worked with my Cytoscape guys to build. It's work in progress, but we're trying to put information in here about how robust a term is. There's different ways, in addition to this noise parameter, there's different ways of assessing the robustness of a term in data. In the original paper in Nature Biotech, we used a bootstrapping-based procedure. So you can imagine bootstrapping the gene similarity matrix at the beginning, and then doing that 1,000 times and constructing 1,000 ontologies, and now aligning them all against each other, or at least against the unbootstrapped one, and looking at how often a particular term is recovered. And one can do that, and it's quite useful, because then, for as an experimental biologist, what you want to do is look at what are all the terms that have the worst alignment score against Go, but the most robustness in bootstrapping. And those are where I want to direct my novel experiments. Thanks for that's a great question. Thanks for forcing me to talk about that. But then I had mentioned to show, you can kind of browse around this ontology online, and this has been quite useful to work with our collaborators on. So if we kind of zoom in here, to this autophagy one, let's just click on a term. So cell proliferation. Due to the low, that's supposed to be a much smaller part of my screen, but it's a low-res projector. So now when I click on that, what I'm now looking at in Cytoscape.js is the actual raw data that supported the inference of the term that I just clicked on, and then one can come down here. Oops. And if you want to know what are each of those interactions and get back to a paper, one can click on those PubMed IDs and so on and so forth. And I think this is also going to be, I don't know if it's in here yet, but it's going to be a good place to talk about, well, we have score, and we give you nothing about what that score actually is, but it's related to the issues you asked about. Oh, I need Teresa, yeah.
So this is really a very elegant set of concepts, and what I'm wondering about is that both your construction and Go, those are very static concepts. Do you envision moving towards the dynamic with this type of analysis, with this type of hierarchy?
So I guess if there was a dynamic part about the vision I talked about, it was the issue of doing more experiments and how you guide more and more experiments as you go through. But I think you're getting on to do you build, what's the right policy? Do we build these for every condition, species? And it's interesting because the Go project has struggled with this for years.
System in terms of using it in the prediction of the dynamical response of the system, or even the cell dynamics.
Right. For me, it's like static genotype. I'm taking a very genetic view of life, right? I have genotypes and I have phenotypes, whereas you're sort of taking the biochemical view of I have phenotypes that are dynamic responses to things, including environments. right? And how do I handle that? And I really, other than have noticing the glaring omission, I've done nothing to address it. What do you think?
Well, I was wondering whether you have as beautiful as it is, I was wondering whether you have a vision how to move in that area.
So we've been talking about, so we've been talking about this. I mean, publishing that first Nexo paper put us into kind of an interesting relationship with the Go team that I'm happy to say was resolved, and we're actually now working together in various ways. And it's interesting to sit on their meetings and hear their discussions, because I mean they basically feel that, for instance, Mike Cherry feels yeast is covered. He's covered every paper in the literature. It's done. Whereas Judy Blake feels like she's got tens of thousands of mouse papers that are uncurated, right? For instance, but then they talk about dynamics, and they talk about was it really the right decision to have 3 branches? I'm no longer sure it is. I think there needs to be one factor, a physical factoring, and you need to functionalize that. That's my vision for what needs to happen. I don't think we need these 3 branches anymore. But again, you're talking about yet another issue, which is, there was originally in the original David Botstein paper, right? It was called A Tool for the Unification of All Biology, and they were very adamant there must be one Go, not one Go per even species, right? It was one Go for all of biology. But then later they've relaxed that a little bit. And this decision also led to interesting consequences. Like you have terms that are like condition-specific terms, like formation of this complex under apomycin, which doesn't make a lot of sense. So anyway, I think you're asking very good questions. Yeah.
Trey, so in your autophagy example and some of the others, you had protein complexes you inferred that had just 2 components, right? So 2 proteins.
Yeah, that'd be the smallest unit. That's not just a gene,
and
right?
So that's just a single physical interaction, which is an edge in your network.
Yeah, and they're here. Like let's look at, so like this thing here, this has got to be what interaction, right? Let's go up. Yeah, right, there's an example.
Right.
Right.
So I guess I'm maybe sort of wondering then, what is really the definition of a protein complex? Why don't we call all edges in the graph protein complexes? Like where there must be some physical distinction, or, I mean, Go added it in there, you said, because there was a paper that supported that complex.
Well, for some of these they're in Go.
Why every pair interaction we could call a complex, but we don't do that. We seem to have some notion that there has to be some larger functional thing going on to make a bigger complex. Where does one sort of draw the line? How do you?
Well, one doesn't draw the line. One just keeps the whole hierarchy, right? But certainly, if you're talking about, are you talking about the gene ontology or how things should be? I guess.
Just in general, yeah.
Yeah. I mean, so what about a complex that has 4 different subunits that all behave, let's just speak theoretically, that all behave exactly redundantly, but they're 4 different genes. The correct thing to do is to merge them all 4 at once into a term. Whereas the interaction graph, it'll come out as a clique, if you're lucky.
But when you look at the graph,
noise aside,
you have many of these approximate cliques, that are sort of.
And that's how it works. And that's exactly how this works.
It's all kind of mashed together in some way. That's really.
No, but that's exactly what this is doing. It's detecting systematically those nested cliques, using an error parameter for.
I guess what's the experimental, characterization that allows one to say that this grouping of 4 is what I should call the complex and not this 5th thing?
Oh, how does one then validate that beyond? So one can do a pull down. So here's an example. So a lot of protein-protein interaction data are shown as flat interaction, pairwise interaction graphs, but the actual experiment is not so. So in an APMS-MS experiment, you have an antibody against one of your proteins, let's say one of those 4, and the other 3 are identified in that pull down. So that's actually a set-based experiment, and we've been showing it as a graph, and then Chink knows there's like papers on this arguing about how you represent this data in a graphical framework, but this doesn't even try.
But Trey, this is a very good example of another problem, because many times you have protein A interacting sometimes with protein B and sometimes with protein C, and many times you have protein A, B, and C being a complex. And at this stage, we cannot distinguish between them, neither experimentally nor computationally, right? And so that leads to a lot of transitive interactions which are wrong, which are actually not.
Exactly.
Okay, but we assume that they are because of the way we collect the data.
Yeah. And the other point I would make is even for there is information that this thing uses or one can use even for a single pair, which is the strength of that interaction. Because keep in mind, you're first integrating lots of information from like the Marcotte way of like 80 different lines of evidence. And so the gene similarity, it's actually in a first degree, every pair of genes has a score. Now I have to admit that the actual computational procedure would be crushed if we took that complete graph as input and did Clixo. So we actually have a first sort of ad hoc thresholding we do. But at least theoretically, you have a complete graph that you start with different weights.
So
yeah, maybe one last.
I want to go back to one of the earlier questions too, thinking about like context. So for yeast, it maybe, I mean, you have different environmental contexts, but it's not like a multicellular organism. So if we think about a multicellular organism, do I have like a liver nexo and a skin nexo? Or how
Yeah, well, this kind of gets back to Teresa's question as well. The Go people would say no, but you can certainly now do it. I guess, I don't know, but you can do it now. That's maybe the right answer.
I think I may have a suggestion. So basically, so far, everything which is in your clustering are just genes. So how about you throw at those conditions as nodes that you learn?
Yeah. I've thought a bit about this, yes.
They come together with the nodes of protein, and they may be able to provide the context that you're missing.
Yes.
By the fact that here is the node with this cluster and comes whatever context.
Exactly. So maybe that's how to do it. Is if everyone heard this, is have terms or nodes that are somehow conditional. Yeah.
I think it's time to thank Trey for the wonderful talk.